Jesper. It's really great to be here. I was saying this yesterday to Kara, the lab. It's actually my world, and I would be considered for the long winters that you're here. So as I'm working, genome instability is actually not only embryonic stem cells, embryonic cells. Okay, right. We're focusing 4 aspects of. We're working on studying to which extent cells are genetically abnormal in culture, at the nuclear genomes, things like that, on mitochondria. Overall, to get an image, does it get in culture? Working as well on trying to figure out how these actually appear in the mechanisms behind this, then take over the culture, because once they're there, they're doing something, they're taking over the whole culture, or not, maybe they're dying. And at the end of the story is, well, okay, how does this have an impact on the behavior of the cells? And especially if we think about differentiating these cells and maybe transplanting them later on in patients. So what can happen if you are using cells that have some form of genome instability? So it's obviously relevant work if we're thinking about what we're doing nowadays with embryonic stem cells or pluripotent stem cells in general. And is that they're making this transition now to the clinic. It took ages to get there. I think we were all very optimistic at the beginning when pluripotent stem cells came into the picture, but now we're finally there. We're getting to the clinic, and we're having this quite significant number of clinical trials ongoing, especially with the retinal pigmented epithelial cells that are being transplanted already in many centers, and they're even proving to be quite, having some effect in any case on the patients. So from a perspective of the genome instability, we know from the work of the past 10 years, and I think it's even more than 10 years now, that human pluripotent stem cell cultures tend to become abnormal. This is a very nice overview from the paper of AMPS, in which they show all the events they measured in different stem cell cultures as chromosome abnormalities. And you can quite clearly see from this overview that there are some of these abnormalities that are recurrent, such as the chromosome 1 gains, the chromosome 12 gains, 17, and chromosome 20. But you have them basically scattered all over the genome. What you can, I think, is remarkable is that it mostly consists of gains. And in fact, there's only one of these gains of which it's pretty clear what is the basis behind this gain. And it's actually this very small common region of gain here that harbors an anti-apoptotic gene that is involved in the pathway which the stem cells activate when you actually dissociate them to grow with single cells, for instance. So this is the only mutation probably that has been well characterized. For the other mutations, there's speculation, like on chromosome 12 you have the NANOC gene, and for the other one, 17, you have a lot of oncogenes going on there. So that's also work we're doing in the lab, so trying to understand why this happens. But I will not be going into this one today. So anyway, why should it bother us that these cultures become so abnormal? You will anyway not use an abnormal culture. If you know it's abnormal, you will not use it to differentiate the cells and transplant them into somebody. But in fact, I think what is very bothersome is that stem cells, they already share a lot of properties with cancer. One of them, for instance, being self-renewal. And another property that cancer has is this genome instability. So it makes you kind of think that maybe this, maybe a little push over the edge will transform your cell and make it potentially dangerous for the patient. So I will be telling you today about our work on a little bit of the work we've been doing on the extent of the genome abnormalities, and later on I will be telling you about our more recent work that hasn't been published yet on how these abnormalities actually are appearing in the culture. So all of this is work of Kurt Jacobs. Kurt Jacobs was a PhD student in our lab. He just recently graduated, so I can't take too much credit for it. So what Kurt did in the first place was studying on the origin of chromosomal abnormalities. And basically the idea is that when you have a cell culture and you do a G-banding or an array CGH to check the karyotype, what you're seeing if you see a chromosomal abnormality, such as any of the ones listed here, what you're seeing is an endpoint. This is where the culture went to because something happened in earlier stages. So, as I said before, there is a whole process of selective advantage behind these chromosome abnormalities that make that this one single event, because it actually does happen, these mutations happen in one cell in your culture dish, this one cell somehow takes over the culture. There's obviously other options, not only that they take over the culture, but the mutant cell can just be carrying some mutation that is very deleterious to them, and the cell would just die, will not survive this mutation. Your cell can have a mutation that will lead to differentiation. So this one is also one that you will not pick up when you do your ACGH or your karyotype, because it simply will be lost out of your pluripotent culture. And then you have the option of a mutation that gives you an improved cell survival, better proliferation, whatever, and the cell is basically very quickly taking over the culture. In our lab, the studies we've been doing in these lines show that in 10 passages, it's enough time to have one cell taking over your full culture and have a clonal culture consisting only of mutant cells. So we were interested in these single events here with the question of how often does this actually happen? How often do stem cells mutate in the culture? And basically to gain an insight on how frequent this happens, and because basically you have these cultures becoming abnormal so often, you would think this is happening very often in the culture, and we wanted just to know more about this. So how do you study single cell events? So you can do metaphase spreads. Metaphase spreads is a very classic method, and you can, in this case, what the authors actually did was counting chromosomes, because maybe, I don't know about you guys, but when I try to make metaphase spreads from embryonic stem cells, they're always pretty bad. They're very ugly, you have a very, very low resolution in your banding, and you don't detect a very small abnormalities. But anyway, you can do metaphase spreads, and what this, in this publication, what they found, embryonic stem cells seem to be prone to losses. And they showed a lot of metaphase spreads with chromosome losses, so a lot of monosomy. This is quite an contrast with what you see, what I was showing you just before in the overview, in which embryonic stem cells actually tend to have gains, right? You can also do fish. There, instead of having an image of the whole karyotype, you will have an image for one locus. If you do what they did here, you really see very badly here, but they did centromere fish. So basically they assumed that if the centromere was gained or lost, that you had a trisomy or a monosomy in the cells. And the bottom line is that with the different methods that have been used, they had quite some differences going on. So the few papers that had been studying these single cell events, they had quite some discordant results, but overall they were saying that there was a lot of chromosome loss. In my eyes, the idea of using metaphase spreads to measure chromosome loss was a bad plan, because in fact, when you're spreading your metaphase, if you get an artifact, it generally is chromosome losses. So in classic cytogenetics, if you have chromosome losses, these are not considered as something real, but considered rather as artifacts. So we were still with the question of, okay, how often do stem cells in culture mutate? So we decided to take an alternative approach, which is doing single cell array CGH. So in single cell array CGH, you collect individual cells from your cell culture, you will isolate the DNA, and in the same tube carry out whole genome amplification, because obviously the DNA content of one cell is not sufficient to do an array CGH. You label the DNA, hybridize this on a slide, and afterwards, where you have your data analysis, you will be able to see relatively small gains and losses. So it's a high resolution method as compared to G-bending. This has the advantage that you can have no gain or losses. If you do this properly, if you're picking up intact cells and you're washing them properly, you cannot have something like a monosomy because you lost a chromosome. So you have the full genomic content of your cell still in your cell, and it's after, when you put it in the tube and you lyse the cell, that you have your DNA free to do all the rest with it. So here you could not have artifactual gains or losses. So what we did was taking two sublines of the same stem cell line, and there were 300 passages apart, so it's a really long time in culture, in fact, that we had there. And we analyzed about 30 single cells for the stem cells. So we took 30 cells of the earlier passage, 30 cells of the later passage, picked up the cells, amplified them, did the array CGH. And what we saw from the results is here you have pretty much the same overview as you saw before for karyotpes. So here it's all the events that we picked up. We saw that the embryonic stem cells, in fact, we found no losses, so no monosomies going on here. We had one trisomy 20, which is one of those that would take over the culture pretty easily. And then you have quite some small segmental gains and losses in the culture. So this is the overview for the 60 cells together. So there's no difference in the early and late passage. In fact, we also did some somatic cells to compare to, and it is quite remarkable that somatic cells in culture, they actually have pretty much the same percentage of chromosome abnormalities, like segmental gains and losses, as the pluripotent stem cells. So here you have a more detailed overview, and I think the most important part is down here. So you have the embryonic stem cells with about 10% of abnormal cells, the somatic cells also with about the same percentages. And in fact, here, I think even it's not statistically significant. So there is no real difference between an early and a late passage of the embryonic stem cells. What this is telling you actually is two things. One is that embryonic stem cells, with their mutations in culture, seem to be much more prone to having a strong selective advantage going on there and a culture takeover by mutant cells and the somatic cells, because somatic cell lines don't tend to become as abnormal as the embryonic stem cell lines. So that's one thing. You have basically the same basal rate of mutation, but the embryonic stem cell lines, they become abnormal much more often. The second thing that it's telling you is that time in culture doesn't seem to increase the rate of low-grade mosaicism that you have in the culture. So most likely, majority of what's going on here is deleterious, and it just lost in the culture. It doesn't survive. And in some events, like you have here the trisomy 20, this will take over your culture. Well, we went a bit further, obviously, to validate the results, because the reviewers did not believe that our single-cell array CGH was that good. So what we did was making clonal lines. So this may be a little bit like links back to the previous talks in which we were discussing whether it's a good idea to make clones or not. Because we made, of one of the lines, we made 7 clones, and then we expanded the clones and did array CGH. Basically with the high resolution array CGH, this one, with the idea that if there was an abnormality in the mother cell, it would then be present as well in the whole culture that we created, maybe next to all the other low-grade mosaicism events, but basically an abnormality that was in the mother cell should be present in the clonal line. And in 1 out of 7 of these clones, we found terminal gains and losses. Just on the side, we've been doing this subcloning now in the lab more often just for other purposes. And it is quite an evil thing to do, because their subclones tend to become chromosomally abnormal. Because when you're cloning, it's your best clone that you're going to pick and expand any further. And often these are containing the chromosome abnormalities. So whether it's a good idea or not to subclone, I would say, but if you look at this, I wouldn't. Or anyways, it's you tend to pick up clonal abnormalities by doing this. So we had to validate this even further, so we went to do fish. And we basically took a bit of a different approach than the other papers, because we had seen all these monosomies also showing in the fish. And what we did was taking 2 random chromosomes, chromosome 1 and chromosome 18, and we took a centromeric probe for the fish and 2 telomeric probes, in which you would have this kind of pattern for a normal cell. And then if you have segmental gains or losses, you would see loss or gain of one of the subtelomeric probes. And in fact, what we did see in the stem cells a lot was this. So as overlapping of the centromeric probe. So for one reason or another, the nuclear structure of these embryonic stem cells in interface, the centromeric regions seem to be co-localizing. So if you're doing fish and you're using centromeric probes, you will be calling a lot of monosomy, which is actually not a monosomy, but just overlap of your signals. So here you have the overview of the results, the two different passages. Here we obviously counted many more cells than with the ACGH because this is cheap and you can screen a lot of cells with this. Is the advantage of fish. We did amniocytes and fibroblasts as well. And here what you do see though is that the embryonic stem cells have a double, more or less, double the amount of abnormal cells concerning the subtelomeric regions. So it does seem that rather than only those big segmental gains and losses, we had quite some gain and loss of the regions close to the telomeres. This was actually out of the resolution of the ACGH that we used for the single cells. But if you think back just a second to what I showed you of the clonal lines, one of those deletions that were shown in the clonal lines is one that was smaller than what we could have picked up with the single cell ACGH. So it kind of all comes down in the, it all fits together. The embryonic stem cells seem to have a tendency of having more gains and losses of the subtelomeric region. So my first take-home message for you is that human embryonic stem cell cultures, and most likely pluripotent, like induced pluripotent stem cells, they form low-grade mosaics of chromosomally abnormal cells in culture. That most of these abnormalities, in fact, are segmental gains and losses. They're not trisomies or monosomies, and that there appears to be an increased instability of the subtelomeric regions. Now, the next question Kurt had to answer was on, well, how do these abnormalities appear? How do the segmental abnormalities show up in the culture? So, chromosome abnormalities, you basically can categorize them in two types. You have your aneuploidies and you have your structural variations, the segmental gains and losses. And aneuploidies have a completely different mechanism of origin than the segmental gains and losses. Full chromosome gains and losses, you will have them because something went wrong during the metaphase. You can have a non-disjunction or an anaphase lagging, by which one of the chromosomes just gets segregated wrongly or just lost altogether. On the structural variations, it generally is because there has been some form of DNA damage, and this DNA damage has been misrepaired in the cells. So our model was, we basically set up this hypothesis in which the culture had something to do with the incidence of chromosomal abnormalities. So you have, in our model, a higher density culture that would lead to some form of stress, metabolite accumulation, or fact that there were not enough nutrients in the medium. And this would then lead to replication stress resulting in DNA breaks, and these DNA breaks then misrepaired and leading to chromosome abnormalities. So we set up a fairly simple experiment, just that we did this in 1 million replicates on 3 different stem cell lines, in which we basically cultured, so this was when we were still culturing the cells on mouse feeder layers. Mouse feeder layers, cells cultured in very low density colonies up to a very high density. So these two extremes are quite unrealistic. So you never culture your cells on this density because it's just simply not effective. And you don't really tend to culture the cells on this kind of density either. So we were normally in the lab somewhere in this range here. And we went on to measure DNA damage with 2 methods. We used immunohistochemistry for a very early marker of double-strand break, and we used the COMET assay, which is a very nice method. I think it's quite fancy and very old-fashioned but fancy. I think you embed the cell in agarose, and you basically do kind of an electrophoresis on it, and you're pulling out the DNA out of the cell, out of the nucleus. And the longer your tail becomes, the more DNA damage you're actually having in the cell. So the DNA, if it's fragmented, it can migrate faster out of your cell. So what did we see? This is the plots for the 4 densities, A, B, C, D, low density, high density. This is tail DNA. This is with the COMET assay. This is with the immunohistochemistry. And here you basically see that the higher the culture density, the more DNA damage you have in your cultures. We went on to measure if this DNA damage actually could result in chromosome abnormalities. And what you see here is the results of single cell ACGH again. 30 cells in each condition. We just did the 2 extremes, so very low density and very high density, basically because the single cell ACGH is very expensive and very labor intensive. And what we saw was that actually at the low density we had no chromosomal abnormalities, which actually was very low. I think it's, just by accident that we didn't pick up any. But in the high density, 33% of the cells carried a chromosome abnormality. And in fact here, I know you don't see the green very well. I should learn to change the slides and make this black or something. So here you have very clearly 5 cells that carry pretty much the same abnormality. They carry this gain of chromosome 1. And this is, if you think back again at the figure I showed you at the beginning, is one of those abnormalities that tends to take over the culture. And the fact that we picked up 5 of the same, it kind of seems to hint at the fact that this abnormality was already busy taking the high density culture. So we went on to analyze the medium to try to find what was causing the genome instability, and there you see glucose levels going down, aspartate, glutamine, but none of them actually reaching a level that you would think, oh, this is critical for the cells, this is way too few. But what you do have is a very drastic increase in lactate and a decrease in pH going on in your culture. So to prove a link between the lactic acid and the pH, what we did was culturing the cells on a very low density, but basically adding medium that would mimic the medium that they would have if they would be growing in high densities. So on cells on low density, we put medium that contained the lactic acid of a high density medium, and we also did the same for pH. So we basically modified the mediums to mimic the high density situation. So what you see then is that this is the mimicked condition, that if you do add more lactic acid to the medium, your DNA damage goes up in the same way as it was going up just by having the cells grown in a high density, and that your pH is also going in the same lines. So you have the acidification of your medium. It basically happens the same thing. You get more DNA damage with a more acid medium. So in a summary image, you have what happens if you grow your cells from low density to high density. If you grow them in low density, but you put the same lactic acid that you would have in high density, and what happens when you do the same for the pH. So basically telling you the pH of your medium is key, the key factor deciding on what's going on with the genome integrity there. So your take-home message for this part is that high-density culture of embryonic stem cells leads to medium acidification by the lactic acid accumulation, and that this is inducing DNA breaks, resulting in chromosome rearrangements in your cells. Now, of course, we were not happy to stay here, and we went to look for solutions for this, because, what's the point of telling you there's a problem if you cannot offer an alternative? So what we did was replacing the medium twice a day. This is very labor intensive, obviously, because normally you replace your medium once a day with the embryonic stem cells. And here you have, so low density changed twice a day, and the same for the other conditions. And what you basically see at conditions C and D is that changing the medium twice a day lowers the levels of DNA damage basically back to the basal levels that you have at very low densities. We went to check if this had any impact on the genome integrity, and this time we used fish, because as I said, the RACGH is very labor intensive and expensive. And if you look at the chromosome rearrangements, basically you have a significant decrease in chromosome rearrangements when you rescue the medium by changing it twice a day. Now, changing the medium twice a day is basically the equivalent of having a slave that is 24 hours a day in the lab, so it doesn't make any sense, it's not practical. So we thought, okay, let's try to buffer the medium better. If acid is a problem, let's change the medium composition. So we're using knockout serum replacement in this setting, and it's a bicarbonate buffered medium. So we decided to add some hESC. So we basically first did a titration, tried to find the maximum amount of hESC we could add. but it didn't help. So here you see the DNA damage, you see the normal conditions and the hESC conditions. And also, even if you look at the pH, the adding hESC to the medium was not capable of buffering the pH sufficiently to go back to the basal level. So this was really not a solution. So now comes the good news for the laminin. So we had to try out something else. We tried growing the cells in a completely different culture system. So this is not anymore using mouse feeders and knockout serum replacement. So it's growing the cells on laminin and using the Nutristem. And here you have, again, so the control, the increased densities in the MEFs. And here you have the laminin. So basically your DNA damage stays pretty much the same no matter on which density you're growing the cells. And when we went to check the chromosomal abnormalities, so this is again by fish, you have no differences between the laminin at high density at low density. So I think it's good news. So you can counter the problem of the medium acidification by changing the medium more often, maybe engineering the medium differently, but definitely not with what we tried, or growing the cells on the laminin 5:1. So to wrap it all up, if you grow your pluripotent stem cells on MEFs at medium to high density culture, so what we usually would do, you end up having a massive amount of DNA damage because of the medium acidification in these conditions. This DNA damage will lead to a low-grade mosaicism in your stem cell culture, and this is the source of the mutant cells that will eventually take over your culture. And you can solve this problem by either changing your density, but it's not very practical because you have very few cells growing in your dishes, refreshing the medium more often, or changing the culture system. So this is part of the team on one of our nice brainstorm weekends. So obviously, I'm not doing this work on my own. As head of department, Karen Sermon, Mieke Gins, and Lise Barbe and Dominica, which are all here today as well. So you can, approach them. Thank you very much. Thank you.